Summary of organic method of DNA purification. In case of organic method, the first step is to cell lysis buffer that is used to lyse or to break the cell membranes. So the first step is cell lysis buffer. So cell lysis buffer is actually to lyse or to break the cell membranes. Nuclei, they are still intact, and pellets of the nuclei are present at the bottom of the tubes. So the first step is by using cell lysis buffer, which break down the cell membranes of the cells which are present in the blood. Nuclei, they still remain intact, and pellet which is formed at the bottom of the tube that contain the actual nuclei of the white blood cells. So these nuclei, they are resuspended, and there is addition of SDS, that is sodium dodenyl sulfate, and proteinase K. So these nuclei, they are resuspended. There is addition of SDS and proteinase K. Both these solutions, they are added to the nuclei. They lyse the nuclear membrane. So in the presence of proteinase K and SDS, they both lyse the nuclear membrane, and they also digest the proteins which are present with the DNA or which are present within the nuclei. Once the proteins, they are digested, the next step is of phenolchloroform. So DNA is released in the solution because the nuclear membrane, they have been digested in the presence of sodium dodenyl sulfate. DNA is released into the solution, and that DNA is extracted with the help of an organic compound which is called as phenolchloroform. So phenolchloroform is used actually to remove the proteins which are present in the solution. So once proteins, they are removed, so we can get the DNA by precipitation, either by isopropanol or by ethanol. So the DNA is precipitated from the aqueous layer because there, when we use phenolchloroform, there are two layers. One is called as aqueous layer, which is present on the upper side of the tube, and the second layer is called as phenolchloroform, which is present at the bottom of the tube. So DNA is present in the aqueous layer, and by adding the cold alcohol or isopropanol or ethanol, DNA can be precipitated. Precipitation by ethanol or isopropanol. DNA can be precipitated or DNA can be washed by 70% ethanol, or it can be dried and resuspended in TE buffer. It can be dissolved either in water or it can be dissolved in a buffer. The name of that buffer is TE buffer. Most of the people, they prefer to dissolve the DNA in TE buffer. So in case of organic method, DNA is purified by using the organic compounds like phenolchloroform.